Hey, good morning, everybody. My name is Mike Ostap. I'm in the Department of Physiology at Penn. And it's my great pleasure to moderate our session, our Future Leaders session today. That's going to highlight the work of Dr. Qing Tang from Malika Lakadami-Ali's lab. And so just a brief introduction to Qing. We know her well here in physiology in the PMI, a very active and important contributor. Who started her career at Wuhan University in China, where she got her bachelor's degree. And then she made her way to University of Vermont, where she initially worked with Gary Ward, doing some really clever work, trying to understand the regulation of a myosin that's important for motility of Toxoplasma gondii. She then stayed at Vermont and worked with Matt Lord, Kathy Tribus, and Susan Lowy, where she did some really important work correlating the biochemical and biophysical activities of myosin 5 and formins with in vivo activity. What really set the work apart was really trying to understand how the biochemical and biophysical properties of these proteins really led to their cell function. Really nice work. She then did some postdoctoral work with Bruce Good at Brandeis, working on isoform differences in the wave complex. And then she's finally made her way to Penn, Department of Physiology, CEMB, PMI, where she's been doing really beautiful work that we're going to hear about today, using advanced microscopy techniques to understand post-translational modifications of microtubules. So with that, welcome, Qing, and we look forward to hearing about your work.
Thank you, Mike. That's very nice of you. Okay, I'm going to start my slideshow here. Okay. Sorry, share the screen. Okay. Can everybody see me and hear me well?
It looks good, and we can hear you.
Okay, great. So, like Mike said, that I have been working on the cytoskeleton for a long time, and I have always been interested in how cytoskeleton is sorted into different functional groups. So today I'm going to talk about my work into how microtubule are segregated into different functional groups. So microtubules are very versatile cytoskeleton network. They're involved in, for example, mitosis, cellular structure, mechanical function, and also often referred as roles for intracellular transport. Well, what you often cannot appreciate from this image on the left is that not all microtubules are the same. And that's because microtubules can be associated, for example, with different proteins, and some of those, for example, can serve as roadblocks. And it can also be modified such that it becomes selective of what gets transported on them. So microtubules are assembled from small tubulin dimers. They're just a few nanometers in diameter, and they can assemble into micron-long scales. And you know, these variations of microtubules are coming from a few different aspects. One of them is the tubulin isotype, and others are post-translational modifications. And they're enriched particularly on the cytosolic tails of tubulins that are often they're sticking out into the cytoplasm. And you can see here this diagram showing you the various different forms of PTMs. They're important because they can directly interface with many different microtubule-associated proteins known as MAPs. And together with the MAPs, they can regulate microtubule dynamics and function. And what has been pointed out in the literature is that many of these modifications are often enriched on the subpopulation of microtubules and segregate them into different groups, and with no known or clear mechanisms. One of the oldest, if not the oldest PTM that we know is detyrosination, which is the removal of the last tyrosine from the cytosolic tail of alpha tubulin. And this modification often marks stable microtubules. Now, what got me interested in this modification is that previously in the academia lab, we have found that detyrosination is often enriched on a small population of total microtubules in a cell. And you can see from this image, and if you zoom in, we see that there's 30% of this total population, but they're spatially mixed with highly tyrosinated microtubules, but they're very distinct. So it turns out that certain organelles become very concentrated on these highly detyrosinated microtubules, in this case, lysosome. And we found that lysosomes are transported 3 times more likely on these microtubules. And autolysosome, which is the fusion from autophagosome and lysosome, are also transported 3 times more likely. So it's possible or highly likely that concentration of detyrosination is a way for the cell to promote the concentration of different organelles for their fusion, and therefore regulating, these degradative pathway. Now, the spatially segregated detyrosination is not is also playing a role in other cellular processes, right? The previous slide is showing one example, but there is evidence showing that detyrosination, they're spatially segregated from tyrosinated microtubules in neurons, mark different microtubule polarity and therefore regulate cargo transport in different directions. And also in mitosis, spatially confined detyrosination helps SAMP-E kinesins to align chromosomes. So what are the potential mechanisms that can make detyrosination become enriched on a subpopulation of microtubules? So we have a few hypotheses in mind. One of them is that perhaps detyrosination is a cooperative process along a single microtubule, or that the enzyme that detyrosinates could potentially bind to some microtubules better. There's also a prior hypothesis in the field that there is a feedback between detyrosination and microtubule stability. So we set out to test these different hypotheses. Now, if you have looked at microtubule in a conventional under a conventional microscope, you'll see that microtubule network is very dense, and it's actually very hard to discern, fine features from them. And this is because how light diffracts. So on a conventional microscope, a point light source will appear as a spot at a focal plane that's much larger than its actual, much larger than its actual molecular size. And if they're far apart, we can discern, for example, 2-point sources because we can locate their centroid. But when they become too close together, 2 spots will appear as one spot at a focal plane. So, and this is the distance, we call it diffraction, the diffraction limit. So in a biological sample, molecules are often packed with distance that are much smaller than those diffraction limit. And when they're all fluorescent all at the same time, this is how you get this blurry image. But using blinking probes, we can just light up sparsely a few molecules at a time so that we can precisely localize them. And through many rounds of iteration, we can therefore reconstruct a super resolved image. And so this is an example of such a microscopy storm, showing you a highly resolved image of microtubules. And so now, if you compare this to the conventional image, we can discern much better these very fine nanoscale features of microtubules. So we started using this to find out how detyrosination is established in cells. And we used a very classic approach in which that we use nicotazole to disassemble all microtubules and remove the nicotazole to allow microtubules to reassemble over time. And then we fix these cells at different time points. And then we use two color super resolution to look at how detyrosination reappear on these microtubules. Now, as a reminder that in untreated cells, we can see detyrosination have these continuous appearance on the microtubule, and then only on the small population of, is concentrated on the small population of microtubules. And when you treat it with treat the cells with a drug, you can see the majority of the microtubules are gone, including the detyrosinated ones. And upon removal of the drug, around 30 minutes, you see the majority of microtubules come back, but the nascent detyrosination appear as small puncta. And they're very sparse. And as time goes on, you see that more and more of these puncta appear, and eventually at later time point, they appear to be larger puncta. So if we measure the size of the puncta and the distance between them, we notice that at the beginning, around 90 until 90 minutes, the length of the puncta almost stayed the same. So the individual puncta stayed the same, but there's a fast fast decrease of the distance between the puncta. Therefore, we think that these small punctas are the building block of the nascent detyrosination, and it's an accumulating process rather than expanding from existing ones. So we also wanted to know what are the nature of these nascent puncta. In other words, how many detyrosination sites there are in each of the puncta. Because in these single molecule localization microscopy, we get pointillistic data. So they're not linearly scaled with a number of targets. However, we can introduce calibration of a single detyrosination site and use those features to estimate the number of sites. Use this feature of the calibration to estimate the size in these images at 30 minutes, 60 minutes, and 90 minutes. And I won't go through the details, but what I'll tell you is that what we have found at early time points, the majority of these puncta contain single detyrosination sites, about 50–60%. And we have some minor population of 2 or 3 detyrosination sites. And as time goes on, these single sites reduced, the proportion of single sites reduced, but the higher number of sites increase. So, the conclusion is that we see these very sparse 1 to 3 detyrosination sites appearing stochastically in the during early washout. Now, these are fixed images. So we wonder how detyrosination occur in live cells. So detyrosination is catalyzed by this group of enzymes known as vasohibins, in brief as VASH. They were recently discovered in 2017. And they appear as cytosolic, as you can see from this EGFP tagged enzymes. And that this has been shown in other studies as well. And in order to look at how single molecules of VASH behave in the live cells, what we did is that we label microtubule network with tubulin tracker. And then we take a snapshot, and then we tracked single VASH enzyme tagged with halo tag. And then we only looked at trajectories that significantly overlapped with microtubules. And what I'll show you is the 2 major motions that we found. One is that the VASH enzymes are diffusing on the microtubule lattice, as you can see that. And particularly in microtubule dense area, they tend to diffuse longer. They also become confined sometimes on a tight spot. So we quantify thousands of these trajectories using MSD, mean square displacement, as a classic way to classify the single molecule motions. As you can see here in examples of diffusive movement labeled in red and confined motion labeled in blue. And we found that approximately half of the motions are Brownian and half of the motions are confined. And here is a snapshot of these movements showing the robustness of these classifications. And we can plot the resident time distribution of each class of motions and calculate their average resident time. And what we found is that in both isoforms, either the confined or Brownian motions have very short resident time, just a few seconds, with the confined slightly longer. Now, this is an important information because in the literature, there has been reported that the K-cat, or the catalytic rate of this enzyme, is about 0.7 per second. That means this resident time allows this enzyme to catalyze 1 to 2 catalytic cycles. So if you remember what I have just told you in this previous slide, that the nascent detyrosination puncta contains primarily 1 to 3 sites, these two results are remarkably consistent. And the two motions that we found, Brownian and confined, we think that because of this slow catalytic rate, the confined motion has just about enough time to allow this enzyme to catalyze detyrosination, whereas because Brownian motion, in which the enzyme travels along microns of microtubules in which there are thousands of subunits, it's likely a surveying or search mechanism for this enzyme. So indeed, we have found a mixed motion in which this enzyme will potentially search for substrate, become confined, potentially catalyze detyrosination, and then search again. We also looked at whether VASH enzyme has certain preferences. So we looked at the trajectories and where it lands. And we don't see particularly regions where VASH doesn't land or VASH excludes. And we also didn't observe any cooperative binding. And further, using different conditions, we can look at whether VASH distinguish tyrosinated and versus detyrosinated microtubules, and we also didn't find there's any difference in their mode of act, in their motions or their resident time. So overall, we think that detyrosination is a stochastic diffusion-limited process. So as I mentioned, that we had a few hypotheses before, but now we're left with this last hypothesis since we couldn't find cooperative mechanism or preferential binding mechanism. So we're left with this feedback mechanism between detyrosination and microtubule stability. So before I go on to the section, I will explain what this feedback mechanism is. So microtubules are dynamic, but their dynamics are different. And that's detyrosinated microtubules are more stable in the presence of MAPs. So I want to point out that the difference between tyrosinated and detyrosinated microtubule dynamics is purely due to the presence of MAPs. They don't have any inherent differences as far as we know. And in fact, recent pioneering work from Antonina Romakak's lab have shown in vitro that if you mix the different ratio of recombinantly expressed tyrosinated and detyrosinated tubulin and make them into microtubules, in the presence of two MAPs, EB1 and CLIP170, you can see that the stability of the microtubule scales with detyrosination level. So again, in the presence of MAPs. So this is a very nice work, but it's in vitro. So in vivo, inside of a cell, we don't really have a way to precisely track the level of detyrosination and microtubule dynamics at the same time. So we decided to use simulation to test the hypothesis whether this feedback could potentially drive the enrichment of detyrosination. So I also want to introduce you to the detyrosination and tyrosination cycle because this becomes important. So As I mentioned, that the primary enzyme that catalyzes detyrosination is VASH, which prefers microtubule polymers. Tyrosination, on the other hand, is catalyzed by tubulin tyrosine ligase, TTL, and it prefers tubulin subunits. So what this means is that detyrosination is stably present on microtubule polymer or microtubules until they're disassembled. And I have shown you in my previous data that detyrosination is a slow process, whereas tyrosination is a fast process, which means that the majority of the tubulins in the cytosolic pool is tyrosinated. Therefore, disassembly and regrowth of a microtubule will recover the tyrosinated state of a microtubule. So in other words, if you stabilize microtubules, promote this polymer form, you will promote detyrosination, whereas disassembly of microtubules will promote tyrosination. Now, I have shown you in this previous slide that detyrosination promotes the stability of microtubule in the presence of MAPs. Here, I'm showing you that stabilization is promoting the accumulation of detyrosination. So this too becomes a feedback loop. And this is the hypothesis we're going to test. Okay, so again, we're using simulation to test this hypothesis, and our assumptions are detyrosination events are stochastic, and then detyrosination on the microtubule lead to its higher stability. So here, how do we define stability? Because microtubule plus ends undergo growing and shrinking, and it has these turning points or transition points known as catastrophe and rescue. And based on previous literature, we define the higher stability as a drop in catastrophe frequency and shortening rates to about 50%. So here is a minimalized demonstration of our 3D spatial temporal model showing 10 microtubules and 10 VASH enzymes. So the green are the microtubules growing and shrinking based on previous literature, and these little flowing spheres are our globular enzymes diffusing in the cytoplasm, parameterized using our single molecule experiments, and they're capable of detyrosinating microtubules with certain probability. Now, in our final simulations, we use 250 microtubules and sub-micromolar enzyme concentration. Okay, so we also have this challenge in that in the field, there hasn't been a quantitative model clearly defining how microtubule stability precisely evolves with detyrosination level on the microtubule. There's no precise molecular mechanism. So we test different models, and one of the models best recapitulated our experimental data. So in this model, microtubules are capable of growing and shrinking. So this is one example. And these it accumulates detyrosination, as shown at these pink patches or puncta. And at a certain point, it reaches a threshold of percentage of detyrosination such that it enters a more stable dynamic regime. So the plus ends will slow down when this percentage is reached, or we call it the threshold. And the way we analyze whether there is detyrosination enrichment is to look at percent detyrosination per microtubule and look at their distribution among the whole population. So if we see a broad distribution, we consider there's no enrichment. But if we see two distinct peaks, we think that this second peak that has a higher average detyrosination shows an enrichment of detyrosination on a subset. Okay, so indeed, when we look at the snapshot of these microtubules and the total distribution, if we don't impose any detyrosination dependent stability, we see detyrosination is broadly distributed among all the microtubules. But if we impose the stability at the threshold when the microtubules are at 20% detyrosination or higher. This microtubule becomes more stable, in other words. And we see a small population that becomes more detyrosinated. Okay, so so this verified that in fact this feedback loop is sufficient to bring out this enrichment on the subset of microtubules. But we further want to understand whether there are key determinants that drive this enrichment on the subset of feedback model. So as I mentioned before that we had this assumption, when detyrosination reaches threshold and leads to higher stability, the stability is defined as reduced catastrophe frequency and shortening rates to about 50%. So this was based on the study from 1993, JCB, Sheldon, and Watworth, where they measured both relatively dynamic and relatively stable microtubules in the same cell. But that was just one study, which provided just the right information for us to model. But if you look at the literature, you will find out that microtubule dynamics vary greatly from cell type to cell type, from study to study. And that's because not only the methods to measure them are different, but also that microtubule dynamics are heavily influenced by the compositions and the concentration of maps, and both the tyrosinated and detyrosinated forms. And their differences are also determined by those the presence of these maps. At the meantime, detyrosination enrichment also different among different cell types. Certain cell types barely have any detyrosination. Other cell types perhaps have, most of its microtubules partially detyrosinated. And of course, some cell types have the enrichment that I have been talking about. That's highly enriched on a small subset. So we want to test whether these dynamic rates between tyrosinated or detyrosinated microtubules can influence the detyrosination enrichment. So, so we went back to the literature, and we went back to this nice quantitative measurement coming from Roman Carrick's lab, in which they have shown that at a different ratio of tyrosination and detyrosination on the microtubule, you can have different growth rate, catastrophe frequency, and rescue frequency. So that means that we can use this as a guide to differ the ratio between dynamic and stable microtubules and test that hypothesis whether these two, these differences between tyrosinated and detyrosinated dynamics can influence the detyrosination enrichment. So here we term that the dynamic are fully, are microtubules are less than 20% detyrosinated, and stable microtubules are more than 20% detyrosinated. So again, at that threshold. And we used 4 different ratios. So growth rate between dynamic versus stable, rescue frequency ratio, shortening rate ratio, and catastrophe frequency ratio. Okay, so with those, in each category, we have 3 different numbers. So for example, here with the growth ratio between dynamic and stable microtubules, we have 0.5, 1, and 2. And same with rescue frequency ratio. And in each of these combinations, we also test 3 different ratios of catastrophe frequency and shortening rates. The way we analyze these permutations is to again look at the detyrosination distribution among the whole population to see if there's enrichment. So if we see 2 distinct Gaussian peaks, very well separated, we give it a high score. And otherwise, if it's not well defined as 2 Gaussian, we give it a low score. So green is highly bimodal, and red is broad distribution. So when we look at these different permutations, we see that the catastrophe frequency plays a dominant role in determining whether there is going to be enrichment of detyrosination on a small subset of microtubules. So what this means is that if we define when the microtubule becomes stable, reaching 20% detyrosination, and that stability allows the microtubule drops its catastrophe frequency for fourfold, it's more likely for that cell to have detyrosination enriched on a small subset of microtubules. So, of course, this is under the threshold of 20% detyrosination and VASH enzyme at 500 nanomolar. And here I'm showing you 2 examples here. These 2 green means by model distribution, you can see that there is a population of low detyrosination level, well-defined, and a population that has a much higher detyrosination level. Okay, so we further tested, along with these different ratios, whether concentration of VASH and the threshold above threshold detyrosination level above which microtubule becomes stable. And again, we test different ratio of dynamic versus stable rates on microtubules. And here, in this lower quadrant, this is what you have seen before, what I just talked about. But in other quadrants, we also have seen that there are different combinations of these rates will allow the microtubule have a small subset that are enriched in detyrosination. So overall, we think that these enrichment can be determined by a variety of combinations. It's not just in one corner of these matrix. So coming back to this feedback loop, what we think is happening is that this feedback itself is sufficient to drive enrichment. So it started as a stochastic process, and by chance, certain microtubules accumulate just enough detyrosination to allow them to become stable. And once they become more stable, that allows them to accumulate a further more detyrosination, and this forms a feedback. And this distribution or the enrichment is determined by a combination of cellular factors. So we think that the rates of detyrosynated microtubules, the rates of tyrosynated microtubules, VASH enzyme concentration, and threshold of percent detyrosination to confirm microtubule stability all play a role in determining the distribution of detyrosination. For example, if we artificially stabilize all microtubules in our simulation, we can drive most of the microtubules having high level of detyrosination. And that has been confirmed by treating the cells with Taxol. And you can see the detyrosination is drastically increased in all microtubules. Also, if we increase VASH enzyme concentration in our simulation, we can also drive a high level of detyrosination on all microtubules. And that also has been confirmed using our overexpressing VASH enzymes in our cellular system in our experiment. But ultimately, I think the rates of detyrosynated microtubules, rates of tyrosynated microtubules, and the threshold to confirm microtubule stability are much more complex, and they're determined by the compositions of maps and to some extent tubulin isotypes. And our model has put forth some predictions that certain combinations of these rates and these ratios will give certain prediction of detyrosination distribution, therefore influencing the cytoskeleton, the microtubule functional segregation. Okay, so I'd like to end my talk by thanking my lab members, particularly Malike, who has been a very brilliant, open-minded, and supportive mentor. And we're a very collaborative lab, and this work has been contributed to by many people. And I highlighted the people that are directly involved in this work. I'd also like to thank our collaborator for running the simulation, particularly Sebastian. And last, I'll leave you with this model. And thank you for listening. I'll take any questions.
Thank you, Qing. That was great. So we have plenty of time for questions related to this fascinating talk, but let me go first. So
Yeah.
you were kind of touching on it at the end about the maps. So, what's clear is that the maps are required for the stabilization of the detyrosinated microtubules. So that would suggest that the kinetics or the lifetime of map association would be a very important parameter. So what do you know about the lifetime of map association, and how does that figure into the simulations?
Yeah, so our simulation doesn't really simulate the maps per se. We're just, having this essentially the rates difference between tyrosinated and detyrosinated. I think the map senses detyrosination very differently. I guess some perhaps are, severing enzyme. They can, chop microtubules apart. Some are, depolymerizing, motors, for example. And I think those probably are relatively fast. Yeah, so I haven't really mapped out all the lifetime. That's a great question. We should look into that and, have more specifics
Okay, great.
taken that into calculations.
Okay, Ben's got his hand up.
Hi, Qing. Thank you very much. It was a really nice talk. I'm hoping you can speak a little bit more about how your studies either conflict or could be reconciled with those from Mary-Jo Wutan's group that came out maybe earlier this year, I think, in JC.
Yeah.
And really, to me, the key difference is that they showed that I think it was VASH1 showed actually quite diffusive behavior along the microtubules, where VASH2 showed much more constrained binding with a longer residence time. And it did some nice work to look at how this correlates with differences in the N-termini of each protein. And now, but theirs was mainly an in vitro study. And so I guess with reconstituted microtubules and purified enzymes. And so I guess I'm wondering, do you just think it's cell type specific cues or map expression that would alleviate these differences? Because in your study, you didn't see any difference, I don't believe, in the diffusive behavior of VASH1 or VASH2. So maybe you can speak to that and whether or not you think it's easily explainable or if there's something else going on.
Yeah, so we thought about that quite a bit because I was brought up by the reviewers. And I think
I'm not the reviewer, just FYI.
Yeah. I think one important components are probably the maps. And I think, microtubules are bound by a lot of maps and I can potentially mask some of the binding sites. So maybe, in vivo that these enzymes don't get to diffuse for that long, 12 seconds or 20 seconds, quite a long period of time, where they potentially, will encounter some roadblocks in the cells. And I think the biggest difference I can think of is the presence of maps, because the VASH binds to the two protofilaments occupying two alpha tubulin. So there's multiple surface. I think many maps will probably occupy, so that can easily bump them off.
Yeah.
And also they have shown that in different ionic strengths that the residence times will change, right? So if at 140 millimolar salt, the residence times are rather long, but when they bump up the ionic strength, that decrease immediately to, 4 seconds. So that's relatively similar. And yeah, so we didn't really see these very prolonged residence time, at least in the population. Of course, we always see some of them having really prolonged, and that's particularly in some microtubule dense region. And one of their micrographs actually strike me is that they see these microtubule skeleton fixed in cells, and they put in these enzymes, and they see these long stretches of detyrosination by one of the isoforms.
Yeah.
And I found some of these long stretches are in microtubule dense area. So I wonder if these, bundled area or crisscross sections where the enzyme can diffuse longer actually get the chance to see more spots to detyrosinate, and that might contribute to these long stretches. And of course, I cannot really tell why there's a difference, when they have these fully reconstituted microtubules. And
well, I think it's a really interesting, I mean, I guess, one experiment, one model it proposes is that indeed the presence, an abundance of maps on microtubules in vivo may limit this potent diffusive behavior, for example, of VASH1 that they show with this rapid detyrosination. But then I suppose you could test this by, many maps are regulated by their phosphorylation status. And so, for example, you could purposely drive the phosphorylation of maps to dissociate them from the microtubules. And then ask, does the diffusive behavior of VASH1 all of a sudden reveal itself with a much more pronounced and more rapid detyrosination, which would be a nice way to at least test that the endogenous presence of maps are a key regulator of that diffusive behavior and detyrosination kinetics.
Yeah, that's a good suggestion. You said phosphorylation, right? I think one of them is MAP4.
Right. Well, MAP4, MAP tau, many of the different maps, their binding affinity is regulated by their phosphorylation status. And there are some common kinases that can regulate these. So I think it would be neat to see if that revealed different behavior in crowded microtubule networks.
Yeah. Okay. Yeah, that's a good suggestion. Thank you.
Yeah. Yeah. Great question, Dan. Okay, AJ.
Oh, hi. A great talk, first of all. So thank you for that. So I just had a question about, so you said in your talk that it appears that the catastrophe rate is the most sensitive, at least determinant of the tyrosination, detyrosination population difference, right? So is that correct? Is that what I understand?
Yeah, so we set that as the, a parameter that we set to simulate, and that makes a difference to drive the enrichment. Yeah.
Right. And so I guess my question was, do you have an insight into, does it suggest, for example, that one might consider, so I'm trying to understand physically why there is a threshold, which is a percentage of the detyrosination state of the entire microtubule. Is it related to some detyrosination near the, plus end, such that the, cap is stabilized or something like that? That might actually lead to catastrophe rates being reduced while other rates may not be as affected. Is that something, or is it something else that you have in mind?
Yeah, so this was indeed a challenging part. We did simulate two different models. One model is that the microtubule plus end can sense the detyrosination state, and then when it encounters the, the first model we tried is the microtubule plus end encounters a detyrosinated patch and it slows down. So that does have the advantage that the more detyrosination that microtubule has, the more stable it becomes. The problem with that model is that the detyrosination or the status of the distribution is heavily influenced by where that first detyrosination appears, and so there's a heavy positional bias, and we could never get enrichment no matter what we try.
Right, but if you, sorry to interrupt, but if you were to imagine that it depends not just on one, but having several in some proximity, then it would be more related to the overall percentage also. So and I think perhaps detyrosination also is sensed by different enzymes differently, right? So severing enzymes can probably sense detyrosination along the way of the lattice, whether it's near the plus end or not. Motors maybe will sense it differently. So there hasn't been a precise model, is because it's a complex problem. There are many different maps sensing the detyrosination probably differently, and we couldn't again, we couldn't get the enrichment from our first model, so we started with the second model based on our experiment suggested, based on the Antonina's study, where she mixed tyrosinated and detyrosinated tubulin to form microtubules, and presumably because there's no defect of polymerization from detyrosinated tubulin, we assume that those distribution is homogeneous. And so that's partially inspired that model.
But again, you know, I think that's still a region, an area that, you know, is still under investigation perhaps by the field, that, you know, is precisely how that evolves with stability. And because of the complexity of the maps, you know, maybe there is a combination of these two models.
Right.
That is just really hard to simulate.
Yeah, that's great. Yeah, thank you.
Yeah.
Tanya Zvitkina has a question.
Hi. So
Hi,
yeah,
Tanya.
I'm just wondering, what specific maps are you talking about? Are there any maps which can distinguish terrazinated and deterrazinated microtubules, and any evidence that there is a feedback? I mean, besides like your
Yeah. So I think
results.
because I think the feedback mechanism is sort of accumulating, is proposed from the accumulating evidence in the field, right? Because detyrosination has been known as a slow process, and then we have shown that also in our experiments. So the longer the microtubule is present, the longer it allows it to accumulate.
Well,
right?
that has been known for a long time. My question is the opposite: how detyrosination stabilizes microtubules.
Yeah, so for example, Katanin, which is a severing enzyme of microtubules, will reduce its severing frequency on detyrosinated microtubules versus tyrosinated, and that has been done in vitro, shown by Antonina Romakak's lab. It has also been shown, for example, that EB1 CLIP170 will drive, will preferentially bind to tyrosinated microtubules and make them more dynamic, right? Their catastrophe frequency will be a lot more higher than detyrosinated ones. There is depolarizing kinesins, such as kinesin 13. They will also preferentially bind to tyrosinated microtubules and drive their depolarization. So there are probably more out there that haven't been, you know, I guess we tested exhaustively, but I think, you know, there are a few known of them that are known.
Okay, thanks.
Yeah.
Thanks, Tanya. Any other questions? Annie, do you have any last things to say? No? Okay, well, we're all clapping for you, Qing. So thank you so much for giving such a wonderful presentation.
Introduction. Yeah,
Okay.
thank you for listening.
Okay, have a great afternoon, everybody.
Yeah. Thank you.